Biology is the only subject where multiplication is the same as division. Stay tuned to discover this fascinating field, one video at a time. In my previous video, I mentioned that I like 10x Genomics and that's why I invested in Illumina. For some of you, it might have been a head scratcher. Today, I'll explain why it is so, and without further delay, let's dive right in. 10x Genomics is one of the holdings in ARKG ETF and have been steadily increasing in weight within the fund since the beginning of the year. What this company does is in analyzing gene expression, whilst the gene editing companies are focused on the genetic material and how they remediate the deviation from the norm. 10x Genomics focus instead on providing the information of what genes are expressed from which cell at any point in time. So why do it? The idea is that having about 30,000 genes does not necessarily mean that every single gene are expressed at any given time within a cell. Only those that are needed will be expressed, and this can be different from that of another cell. We call this differential gene expression. The significance of this was observed in a curious case of cancer patients. Specifically, it was observed that a set of genetically identical twins may not have the same genetic diseases like cancer. You would think that if a pair of twins have the exact genetic makeup, then wouldn't they suffer from the same genetic disease? If cancer strikes one of the twins, wouldn't the other also suffer from it? Upon further study, it was observed in this phenomenon that the genetically identical twins lived apart from each other for a very long time, and the environment is very different for each twin. So one possible explanation is that the environment influenced differences in gene expression, and it is this gene expression that led to the development of disease in only one of the genetically identical twins. And this is why the information about gene expression is so important besides the genetic makeup of the cell. So what's the mechanism behind this? One of the possible mechanisms is based on the concept of epigenetics. You can click on the I button appearing above right now for a quick refresher before proceeding. Epigenetic control of gene expression uses the concept of methylation of nitrogenous bases, and this in turn could be due to the presence of a chemical messenger in a certain environment that will bind to a receptor on the cell membrane. In a different environment, the chemical messenger could be absent. The binding of the receptor activates a signal transduction pathway, the last of which could be a DNA methylating enzyme, and what gene was once expressed when methylated would stop gene expression. But I'm not done yet. As a biology tutor, my smart students may ask me if there's any situation where a pair of genetically identical twins in the same environment may have a different disease outcome. Great question! The answer is yes, and let's explore it. As humans, we are diploid organisms having a copy of genetic material from our father and the other copy from our mother. So for every gene, we've got two copies. Typically, the normal copy of genes are represented by an alphabet. Let's say A, which would be capitalized. And another version of the same gene was produced during evolution, a result of mutation that reduced the function. So let's represent it by a small a. And because of the reduced function, this would lead to a genetic disease. Big A has dominance over small a. So in this genotype of big A, the small a is not observed physically, since big A is present and in control. By right, a person with this genotype should not suffer from the genetic disease. Only a person with this genotype would suffer from the disease. Now, let's return to the pair of genetically identical twins. Let's say they are of the genotype big A, small a for a particular gene, and the environment causes methylation to occur. But in this particular gene, only one copy is methylated. So for one of the twins, the methylation occurs to a small a and the big A is still unmethylated, so things are fine and dandy. However, for the other twin, the big A is methylated instead, and the small a copy is expressed, leading to disease even though both have the same genetic makeup and living in the same environment. These therefore are the reasons why profiling the gene expression is very important. And 10x Genomics is at the point where its technology can easily identify what's being expressed in a cell, and this information could help the scientists looking into the cell biology to answer questions about disease. So the next question to ask obviously is, how does 10x Genomics technology work? Let's find out. When the cells are isolated from an individual, they are separated and suspended in a solution and then fed into the 10x Genomics Chromium controller machine. During this time, the cells are associated into a gel bead-in emulsion particle. The gel bead is unique and there are many different gel beads to start off with. In doing so, each gel bead is identifying the gene expression profile of one cell. In addition, the gel beads with the cell is suspended into oil, which acts like a partition by compartmentalizing the reaction that is about to happen. And within it also contains all the reagents required for the subsequent reaction. One of the reagents is the enzyme reverse transcriptase, which reverse transcribed all the mRNA expressed from the genes to DNA. This is known as cDNA because the sequences are derived from the mRNA. So how does reverse transcription work? Each bead has got millions of tiny strings of nucleotides. Each of them on one bead has a unique barcode representing that bead. To one end of this string of nucleotides contains repeating thymine bases. This will be bound by mature mRNA, which has a poly-A tail via complementary base pairing. Once done, the reverse transcriptase will start synthesizing the cDNA from the different attached mRNAs. These beads are then recovered in the microfluidic cells and sent to the short-read sequencers. The sequencing data is then fed back to the TXG Genomics software to identify which genes are expressed and even how much expression is occurring. For example, an active gene can be expressed into mRNA hundreds of times, and this can be counted during sequencing. A less active gene will have less mRNAs and would therefore be counted less. If cells are isolated from different organs of an individual, or between different individuals, the differences in gene expression can therefore be compared. In my previous bio-nanogenomics video, I mentioned that company cannot resolve tumor heterogeneity, but TXG Genomics can. And the reason is, it is looking at what is occurring in every single cell. And TXG Genomics is way ahead here. I know by this time, the question must be killing you. Why did I invest in Illumina when I like TXG Genomics so much? Remember in the previous section, I mentioned that the beads have to be recovered, and each of these beads contain different strings of the sequences that are derived from different mRNA. Yes, those, they have to be sequenced. Since the mature mRNA are much shorter, this would be perfect for the short-read sequencing that Illumina is famous for. In the establishments that's already using Illumina's machines, this will increase the use case scenario and the demand. Therefore, when TXG Genomics continue to grow, this will further add onto Illumina's bottom line. Consider the market that's generated from Grail's cell-free DNA screening for cancer, as well as the gene expression studies using the TXG Genomics machines, as well as the bread and butter sequencing that Illumina already does, will make them the true king of the north. Dethrone the king? Not anytime soon. However, I did not directly invest in TXG Genomics because the company's profit margins are capped. This is because they have been successfully sued by Bio-Rad as well as the University of Chicago. As a result, they have to pay a $24 million penalty as well as a 15% royalty on sales. I can't help but wonder about ARKG's thinking when they dumped Illumina and purchased position in companies like Exact Sciences and TXG Genomics, which contribute to Illumina's bottom line, and in the same time miss a huge Illumina bull run. Let me now talk about some protein sequencing companies that you have requested in the comment section in the previous videos. While there's a strong argument to support the use of TXG Genomics technology, protein sequencing is one step further down and does not add much more value. Let me explain. After gene expression, synthesis, and subsequent processing, mature mRNA will be read and translated in the ribosome, leading to the production of proteins. Most of the mRNA will be translated into proteins, and there are only a few situations where they are not. Firstly, when a cell undergoes division, and second, when fertilization is occurring. Finally, when there are already sufficient numbers of proteins. The difference between the number of mRNA translated to protein is relatively small compared to the genes and mRNA. Therefore, I currently do not see much growth opportunities since the need to look for proteins is not as urgent as that of DNA. So companies like Seer and Quantum-Si is not in my biotech stock radar as yet. This section is for the Watch Till the End gang as a thank you for tuning in and for watching all the way through. Next week, I'm going to talk about my first biotech stock investment, Beam Therapeutics. There are a number of new developments to catch up on. In the meantime, you can catch up on my past videos about Beam Therapeutics here.